All right. Hi, and welcome to the next webinar in our Plantae webinar series. My name is Jason Padilla, and I'm your technical host for today's webinar. Next slide, please. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all our ASPB members who are attending today's webinar. Your ASPB membership dues help support making this webinar possible. If you're not already an ASPB member or have not yet joined or need to renew your ASPB member, you can do that today using the discount code %10 to receive a 10% discount on your registration. ASPB members get early access to this webinars, and you can learn more about ASPB and all the many opportunities we provide at aspb.org. Next slide, please. In addition, this webinar is recorded, and we have so many others Plantae webinars that we've done in the past. So if you're looking for new or different plant science related content, you could watch them on our Plantae YouTube channel, and so be sure to check those out. And this webinar, again, will be added to the library of our previously recorded Plantae webinars. Next slide, please. Registration for a plant biology conference is already open. We are currently using essentially our advanced rates, so you could get still pretty good discount if you're looking for a plant science conference to attend, especially this summer. We have plant biology registration open. This year, it's taking place in Milwaukee, Wisconsin on July 26th to the 30th. It is a very uniquely positioned conference that brings together members of the plant science community that are coming from all over the globe. And it's a great chance for you to acquire knowledge, network, and develop new skills necessary for you to help really our quest or our goal to advance plant science, and it's open to all career levels. You can learn more about plant biology at plantbiology.aspb.org. Next slide, please. Before we get started, I'd like to go over a few housekeeping items to make sure that you get the most out of your attendance today. If you are experiencing technical issues, let me know about those using the Q&A box, or you can email me at jpd@aspb.org. If you have questions for our guest speakers, please add them in the Zoom Q&A box again. Our moderator will be sharing them with our presenter during the Q&A section of this webinar. If you have trouble connecting or need to leave the webinar early, as I mentioned, this webinar is recorded and will be made available on our YouTube channel. And so you could rewatch and also share this with any of your peers or colleagues or people that you know that might appreciate or would benefit from the content of today's webinar. So again, be sure to follow our YouTube channel and check out all our many other Plantae contents. All right. Now I'm going to turn it over to our Plantae Fellows who are moderating today's webinar. And so with that, take it away, Plantae Fellows.
Thank you, Jason, for helping us make this webinar like a reality, and a very warm welcome to everyone who is present for this particular webinar. So today's webinar is going to be moderated by 3 Plantae Fellows, including me. I'm Aditi, and Kes and Gabriella. So basically, like, the main issue like currently is like you have climate change going on. Like there's so much issues with like lab. How do we make it more sustainable? And given that we are plant scientists, there's no dearth of greenness around us, like whether be it in lab spaces where we have those tiny Arabidopsis seedlings or duckweed growing on, or even in the field where you have like crops like corn or maize growing in the field. But the issue is, do you think about like whether like being, in addition to being like working on green spaces, is our research work truly green or not? And I know I do think about it all the time whenever I see a tons and a countless number of pipette tips or gloves being discarded every single day, or like so many big machines being hooked up and taking up like being energy guzzlers. Given that you guys are attending the seminar, so I'm sure that you also have similar questions in your mind. And to alleviate to answer these questions, we Plantae Fellows have actually developed this particular webinar and put together a stellar cast, which have experience ranging from developing a particular entire not skill set, but entire regime of how do different organizations develop a program so that their research work is becoming more green in nature, and even two research investigators who will share their experiences how to execute these major programs that are being developed into their day-to-day life so that we can make our research work more green as well. So next slide, please. So as I mentioned, for today's particular webinar, we have 3 excellent speakers. So we have Caitlin, who is a sustainable research officer at LEAF. And then we will be listening to Dr. Lise, who is a senior lecturer at plant sciences in plant science department at University of the Free State in South Africa. And finally, you will listen to Dr. Malia, who is a principal investigator at Donald Danforth Center, who works a lot on temperature stress responses and developing high throughput phenotyping platforms for doing analysis. So with that, I will give the stage to Kes, who will be introducing our first speaker. Oh, I'm sorry. So yeah, just to get a brief overview of what you'll be listening into this webinar is, as I already mentioned, the general introduction of the webinar. And finally, later on, we will listen to the speakers talking about their own experiences, how to make the research more sustainable in their research program. And then we'll have a question and answer session, which is towards the end of when all the speakers have spoken. So we'll have around 10 to 15 minutes where the speakers will answer specifically some specific questions which the audience will have. And we encourage all the audience members to actually type in your questions in the Q&A box throughout the webinar. We'll be monitoring it, and the speakers will answer those questions towards the end of the webinar. And finally, we will finish up with some concluding remarks. And with that, I will give the stage to Kes, who will introduce the first speaker.
Hey, hi everyone, I'm Kez. I'm a second year PhD student at the John Innes Centre, and I'm one of the moderators, as Aditi said. And I'm using microscopy and molecular biology in order to resolve how floral organs develop, essentially, so floral organ morphogenesis. So starting my PhD was really a wake-up call in how easy it is to waste plastic and energy. And I really started to learn more about how to combat this through the work that LEAF's doing with my institute, the John Innes Centre. And so with that, the next slide, please. I'm really honoured to announce our first speaker, who's Caitlin Broadbent. So as Aditi mentioned, Caitlin is a Sustainable Research Officer over at LEAF, which is based at University College London. And LEAF is a framework which is used worldwide in over 130 institutions in order to help lab-based researchers improve their environmental impact. And Caitlin's own background is in molecular biology, so she has extensive experience in wet lab research. And so it will be really great to hear about how her personal experiences and how LEAF implements strategies to overcome the conflicts in research and environmental impact. So I know she'll introduce more about how that works. So with that, please take it away, Caitlin.
Thank you very much. So I'm going to share some slides. So bear with me a second. And what I am hoping you can see my slides. There we go. Cool. So as Kez mentioned, my name is Caitlin Broadbent. I'm a Sustainable Research Officer at University College London. So we're based in the UK. And my role is helping all of our external organisations with the LEAF programme. So UCL created LEAF, I think, in 2018. we do LEAF within our labs here, but we also license it as a program to other universities and organizations across the world. And it's my job to help people with that and also with kind of sustainable research in general. So today I'm going to talk about kind of the impact of laboratory research with a kind of focus on the wet lab. What is LEAF? The idea of putting LEAF into action. So LEAF is a framework that you can sign up to, but what can you do without signing up? And then some of the networks that are available that you might like to join. So how bad is lab research? So this is a really nice report from the Royal Society of Chemistry published in 2022. And I'm just going to kind of point out some of the areas of the lab that have an impact in terms of energy waste and carbon. So when we think about energy use in the lab, there's probably some really obvious pieces of kit that you're thinking of that are on all the time. They use a lot of energy, but some kind of, I don't know, surprising ones can be things like lighting. So lighting in the labs can contribute anywhere between 10 and 28% of electricity use. You've also got your kind of analytical instruments that can use kind of over 690 kilowatt hours per year of energy. Drying ovens, if you need any of your glassware or pipette tips coming out of the autoclave dried, that's going to have an impact. And fume cupboards. So these can use 3.5 times more energy than the average US home. When we think about water, if we're using anything like deionized water or ultra pure water, every time you make a litre of deionized water, you're wasting 2 litres of water because of how the process works. And when you then make ultra pure water, you're also usually using extra consumables and UV as well. And then if you're using water for things like cooling reactions, you could be using up to 15 litres per minute if it's just single use. And then the final one, this is a really big one, is plastic waste. So there's an estimated 5.5 million tonnes of plastic waste, which is about 2% of the global plastic waste just from biological, medical or agricultural research. And I'm sure anyone that stepped into a lab that has single-use plastics has noticed the amount that we put in the bin. Now, a lot of this is often incinerated at high temperatures with little opportunity for energy recovery. So it really does have a large carbon impact. So where does the LEAF program come in? The question was, okay, we want to make a change. How do we do that? So LEAF was created. So LEAF is the Laboratory Efficiency Assessment Framework, and it is a framework. The idea is that people from a grassroots lab level can make changes in their labs and to the processes that they use to be more sustainable. So LEAF is a combination of kind of like 4 aspects: the framework, which is a set of criteria, calculators, so you can quantify the impact you're having with the changes you're making, and you can also put a number on things like energy use in your lab, a toolkit and resources, so things like case studies, documents, guides, and then user engagement as well. So we know it doesn't just stop at here's the framework, it's also, okay, how do we get people to do this? These are some of the examples of criteria, and this is kind of what I'm going to focus on a bit today. So we've got different categories, everything from people to equipment, and then we've got 3 levels, so bronze, silver, and gold. And at each level, we have set criteria that people have to complete. And when they've completed that range of criteria, they can apply for a LEAF certificate. And just to make sure that our certification is robust, we require an in-person audit. And from this year, we are offering auditor training so that audits are done to an ISO standard, because we know a lot of people do at institutional level the ISO 14001 for environmental management systems. So I'm going to talk about kind of how you can use these 15 criteria to take action in your lab now without having to sign up to any sort of program. The first thing we'll kind of have a look at is reducing energy. So for example, one of our criteria is turning off equipment and lighting when it's no longer needed. How do we do that? How do we put that into practice? We're also going to look at things like fume cupboard sashes and keeping them closed when not in use. So this is kind of energy. What can you do from an energy perspective? The one thing you can do is use stickers and signage. So for example, you might have equipment with a computer and monitor associated with it, some of which cannot and should not be shut down. Certain things like NMR, SEM, TEM, you do need it to stay on all the time. But you might have a piece of equipment that you can shut down. And so stickers that really very clearly state whether you can or can't turn off a computer or monitor can be very helpful, especially for new people in the lab, not necessarily worked with that equipment before. It's very clear as to whether they can turn off or not. So you could have a thin sticker like this on your monitor, for example. You can also have always switch off, don't switch off, ask before switching off stickers on things like ultra low temperature freezers, incubators, water baths and heat blocks. So this is really clear saying, look, you can always switch this off once you're done with it, as that kind of visual reminder. And then what if people don't like turning off equipment because they think it takes absolutely ages to start back up again? Well, you can let them know this is how long it takes for you to start this piece of equipment up, ready for it to be used. So for example, in a lab I used to work in, we had a UVP for imaging gels. In my head, it took hours to start it back up, lots of software issues, but actually it was only about 10 minutes. So now, you know, we could have added a sticker like this saying this piece of equipment takes 10 minutes to start up and be ready to use. And if you've got something that needs to reach a temperature, you can say this water bath takes 20 minutes to reach 37 degrees. That means people can turn it on in advance, but then also not turning it on 2 hours in advance if it only needs to be turned on 20 minutes before. The other thing you can look into is timers and energy monitors. So it's kind of two types of timer. You could get a booster and an on-off. So some people find that their water baths are left on overnight. If you were to put it on a timer and set it to start at 8 a.m. and turn off at 6 p.m., you'll reduce that kind of 24-hour time frame to more of 12 hours. So you can make changes in the lab. Most timers are not very expensive, but I do realise that obviously not all universities and institutes have the money. And this is kind of a photo of all these things in practice. So here's a piece of equipment, a computer and monitor associated with equipment over here. And we're saying it takes 10 minutes to be ready to use, and a green sticker here, you can just about see it saying always switch off. And it's little things like this. It gives people the confidence to go, okay, I do know I can switch it off and I'm not going to break it. But I also know I shouldn't shut that off because it does need to be on all the time. So what about fume cupboards? When we have a variable air volume fume cupboard, the height of the sash becomes important. So when you raise the sash, the fume cupboard is drawing in more air, therefore it's using more energy. As you shut the sash, it's drawing in less air because it knows it doesn't need to pull as much on its fans, so you reduce the energy. A lot of people are still in the habit of leaving their fume cupboard sash open, so it might be up or to the side. And this means if you have a VAV fume cupboard, you are wasting energy. So Cornell University looked into how we could change that behavior. And it's quite easy. You just introduce a little smiley face. So if you take a smiley face, you put one part on the stationary part of the fume cupboard and the other on the bit that moves. People like to put them together. And what they found through their research, they compared a set of labs that had been given this information, the smiley face, and a set of labs that hadn't. It ended up leading to a 52.8% reduction in instances of the sash being left open. So this is a really nice paper that looks at behavioural change in the lab. And then they kind of went a little bit step further and they compared different floors to each other to see if that would then again encourage people to start closing the sash. And so they used smiley faces. So the first floor, you know, they were doing quite well at the start, a little bit off at the end. And they kind of noticed that in the unoccupied state, the number of times the hoods were left open dropped by 19% during the experimental period. And then after that, it stayed at a drop of 42.5%. So simple things like these signage, which you can print and cut out, we've got the link on our website, can make a massive difference. It's also really good for health and safety and protecting yourselves in the lab. So that's just a couple of energy options. Now I will talk a little bit about cold storage. So a lot of cold storage is to do with energy, but it's also about kind of sample management as well. So if we take an idea around procedures that are in place in case cold storage equipment breaks down and cold storage is maintained, we can look at how you could do that. So why is this important? This is the impact of ultra low temperature freezers. And by that, I mean freezers that go to -80 or -70 degrees Celsius, which is -112 degrees Fahrenheit. We know from research that actually the carbon associated with ULT freezers, 91.8% of that is during its use phase. So we can control 91.8% of the carbon impact of our ULT freezers. The average ULT freezer set to -80 can consume anywhere between 9 and 20 kilowatt hours per day. If you set it to -70, you can save up to 28% of energy. And we also know a lot of the older ULT freezers, so if you've got one that's over 15 or 20 years old, you might have a refrigerant in there doing the cooling that has a high global warming potential. For example, R404A has a GWP of almost 4,000 times that of CO2. So that's kind of the ability of of the chemical to retain heat. The other thing we want to avoid is freezers that look a little bit like this. So if you were to pull the door open a little bit too harsh, that box in the front there, the yellow one, that's falling out, all of your samples on the floor. You can't really get to the back of that freezer. So you've got samples in there that you're not using. You might end up having to buy replacements. So overall, looking after our freezers is important, not just for energy, but also for how we do research. So really simple things. One is maintenance. So things like having a information sheet on the front of your freezer with a list of maintenance activities, when it was last done, when it's next due, and who's doing it. And also having things like frequency. It's also really useful to have this kind of key information on your freezer as well. And if you do have an ultra low temperature freezer that's set to say -70, it's good to put the set temperature on the information sheet as well. So that if someone comes along to your freezer, says, oh gosh, it's at -69, is that a problem? Thinking it should be at -80, they actually know it, no, it's meant to be at -70, it's fine. You can also use this for health and safety purposes, so you can kind of indicate if you've got hazard group biological agents, if you've got any sort of chemical hazards in the freezer that people need to be aware of, and you can add what to do in the event of a breakdown. The quicker we can move items to another freezer, the safer everything is. Maintenance is really critical. The reason for that is we know that if you don't clean your filters and you don't remove the ice around the seals, the door doesn't seal properly, and you could be using 27% more energy than you need to keep your ULT freezer at its set temperature. The other thing you can do is have a very basic inventory. So ideally, you would have inventory software, but some of these can be quite expensive. You would then consider maybe an Excel spreadsheet, but even having something on the door saying where your items are can be really helpful. This is one that we put together for a backup freezer, so it's only something that's filled in when needed, but it's got things like, racks and shelves. The reason that's really useful is because it takes a lot less time to find a sample if you already know where it is. That also means your door is open for less time and your freezer is getting warmer less, so it has to cool down less. For example, it takes 133% more time for a racked ULT to warm to -50 from -80 than an unracked ULT. So this is really good for breakdowns. If you have a breakdown at 3 a.m. in the morning, you've got longer to sort it out if you've got a racked freezer. And then also, if you've got that door open for 5 minutes compared to 1, it's going to take over 200% more energy to reach that set point of -70 or -80, which is a lot. At the same time, your samples are going to be at risk. So this is why just really simple documents that you can have on the front of a freezer can be very helpful. So now let's think about plastic. I've used a lot of plastic in my day. It's so sad putting it in the bin. So how can we reduce single-use plastic? A couple of questions you can ask yourself. Do you need to use it in the first place? So for example, if you're in a teaching lab, you generally, as a technical member of staff or an academic, you'll know exactly what's happening in the lab. You can assess whether people need to wear gloves or not. But the same goes for research. Do you need to wear gloves? Do you need to use a piece of plastic? Can you choose a different material? Now, there are some activities that don't relate to different materials. There are certain chemicals you have to use plastic for, but can you replace it with something else? If you can't, can you reduce the size? Instead of using a 2 mil or 1.5 mil microfuge tube, can you use a 0.5 mil or a 0.2 mil, so a PCR tube? And if not, can you change your method? So can you reorder the steps to reduce the number of items you're using? So one really good example is in a PCR reaction, if you're genotyping, if you add the master mix first, you can do that with one pipette tip and then add the DNA. If you add the DNA first and then the master mix, you have to use a different pipette tip for each of those. So just by changing the order of something, you can reduce your plastic use. The other is wash and reuse. So a lot of people kind of say, what is the benefit of wash and reuse? Is it better? Is it worse? Does it use more energy? And this is a really nice paper that actually looked into this. So they compared single-use plastic, reused plastic and reused glassware, and they looked at a production, use and disposal, and they found it was better to wash and reuse even if you need to autoclave the item. And so there's a really great group at Sir William Dunn School of Pathology, the Green Group, and they are doing just that. So they are taking their Falcon tubes, they're decontaminating them, washing them in a dishwasher, autoclaving them, and then putting them back together in a biological safety cabinet so they stay sterile. So it is possible to reuse plastic and reuse glassware in environments where sterility is key. We also have a really nice paper from the University of Edinburgh, and they looked into how they could reduce their plastic waste in a microbiology lab. So another space where it's really important to consider the risk of contamination. And just by changing the method that they used to do the same activity, they went from 36 agar Petri dishes down to 1 agar Petri dish. Now, if you do this once, that makes a decent difference. But if you're doing this 100 times a year, that's a lot of plastic that you are going to be no longer using. So what if you don't want to do LEAF? What if you don't work in a wet lab? There are other programs you can get involved in. So the Software Sustainability Institute has a program called Green Disk. This is for computational researchers. It is the Green Digital Sustainability Certificate, and it's a framework, a set of criteria that you can follow on software and hardware. If you have a lot of ULT freezers, a lot of cold storage, liquid nitrogen in your lab, the Freezer Challenge is a free challenge that runs from January to July every year, run by my Green Lab. And it kind of just goes above and beyond that cold storage aspect. You get points for doing innovative and novel actions like high density storage. And then you've also got Green Impact. So this is run by SOS UK, and it kind of looks as a framework for office spaces. So if you have more dry labs, computers for students and teaching, this can be very useful. And in terms of getting started, the main thing is you're not alone. There's loads of people out there that are making changes in their labs. We have got Lean in the UK. There's also the International Institute for Sustainable Laboratories. Green Your Lab is an online discourse forum. There's lots of information out there for how you can get involved. So if you're not wanting to or not able to sign up to a formal program, you can still connect with other people that are sharing tips, tricks, best practice, resources. And we have loads of things on our website that you can access for free as well. Thank you for listening. If you want to find out more about LEAF, you can go on our website. Our website's quite long, so if you search UCL LEAF, that's quicker. We've just started a LinkedIn, so it's quite barren, but we'd love to fill it if you want to share any best practices. And then we do have Blue Sky and an email as well. Thank you.
Thank you very much, Caitlin. That was a great talk. Very interesting to hear what you are doing from LEAF, and also have some ideas on how can we make our research more sustainable. I'm Gabriela Doria, so I'm also a Plantae Fellow for 2025. I'm very excited to be here in this webinar, to have the pleasure to organize with my fellows Aditi and Kes. I'm also worried about what we do in the lab, also thinking about how could we make it cleaner, how could we make it more sustainable. I started kind of my activism on sustainability in the lab during my time as a PhD at the University of Cambridge. And that's where I met Lise, our next speaker. So I'm very excited to hear from Dr. Lise Joubert, a senior lecturer at the Department of Plant Science at the University of Free State, who's focused on anatomy, micromorphology, pollination biology, Evo-Devo research also in several taxonomic, very interesting families, a very interesting flora of South Africa, and how she does integrate sustainable practices in her lab and in her work. So welcome, Lise, and please go ahead with your talk.
Thank you very much, Gabriela. It's nice to be here. So as Gabriela mentioned, I am from the University of the Free State in South Africa. So we have some interesting challenges regarding green lab practices. So in my talk, I'm going to talk about my own lab, but I'm first going to give you a bit of perspective on the situation at our university and in the country. just to create a context. So strategies for having greener lab practices would include reducing energy and water consumption, reducing waste, recycling, and proper disposal of hazardous waste. And all of these Caitlin touched on in her excellent talk earlier. So South Africa is a country that's mainly dependent on carbon-based electricity or energy. At least that has been the situation throughout most of our history. But just under a decade ago, some challenges arose with our national power supplier, and the situation arose where we experience load shedding. So load shedding means the grid is under pressure, there is not enough electricity to satisfy the demand, and depending on the day and the situation, you could experience anything between 2 to 10 hours without electricity. So obviously this is not great for running a research lab. It creates lots of problems, and having the energy go down and come up again is also very dangerous for much of our expensive lab equipment. So the university has recently invested in solar plants on all of its campuses. There are both ground-mounted and roof-mounted solar plants. So the one you can see here is on the Bloemfontein campus where I am stationed. That is a ground-mounted solar plant. Here you can see a roof-mounted solar plant. So the one on Bloemfontein campus generates about just over 3,000 kilowatts peak. On our Kwakwa campus, which is much smaller, the solar energy is about 900 kilowatts peak. And on the South campus, where there are no research laboratories, there are just lecturing halls and offices, about 700 kilowatts peak. And then Paradise is an experimental farm. So the energy consumption there is mainly for the animal husbandry. So since installing these solar plants, the university's energy consumption has decreased with about 14%, even though the university's lab area and office areas have expanded. And it has been calculated that our carbon emissions have been reduced from 0.115 carbon dioxide per square meter in 2013 to 0.088 carbon dioxide per square meter in 2022. So this is excellent, and there are several other initiatives taken by the university to reduce energy consumption. Air conditioning systems are centrally controlled and switched off from time to time, except for climate control systems in the plant growth facilities, which are kept on. And then in the lab, of course, equipment is switched off when not in use and usually unplugged to avoid the damage by power surges caused by load shedding. And then having equipment that's energy efficient is very important. But as you can see from this picture, much of the equipment in our labs is still quite old. So we have a program to gradually replace the older equipment with more energy efficient equipment. But that is a very slow process. So once a year, you get to apply for funding to upgrade equipment or replace equipment, and that is very competitive. So it's not always possible, but our Kwakwa campus recently had a great upgrade in their lab. They had new equipment installed and their lab space also renovated. So there is some progress in that regard. Now, South Africa is an arid country, so saving water is high on the agenda here. So on campus, they have established water-wise gardens. They are using grey water for irrigation. Rainwater harvesting systems have been installed with all the major buildings, office buildings, and student residences. And then in the restrooms, public restrooms, push-button taps and waterless urinals have been installed to reduce water usage. Now, as Caitlin mentioned, when we use deionized water, for every liter of water we use, 2 liters are lost or wasted. So in our lab, we do not have deionized water on tap. It is provided in these containers. So students have to take this container up to the DI room to refill it when they need it. So they work sparingly with the water because it's a job to refill the container. And all the runoff from the DI process is captured in a wastewater tank, which is then also used for garden irrigation. So it is not discarded immediately. Reusing and recycling has proven to be a challenge. So a couple of years ago, we had a big drive to have recycling on campus. But the decision was to reduce costs and use the existing waste bins on campus and just adding lids to them. So color lids and labels. This bin is for plastics, this bin is for paper. It was a really great idea, but it didn't work at all. First of all, I think there should have been a bigger effort to educate the students and the staff about this initiative and to make them aware of the need and the importance for this. And secondly, maybe more money should have been spent on providing more bins in convenient places, because the students ended up just throwing whatever they had in their hand in any bin. There was absolutely no separation of recyclable materials from regular waste. So that whole initiative was dropped, unfortunately. So I hope we can reinitiate it at some point. So in the lab, we recycle everything we possibly can. So our lab is an anatomy lab. So obviously we use lots of microscope slides, SEM stubs, silica gel for drying, and all of this can be reused. So our microscope slides, wherever possible, are cleaned. Even the cover slips are sometimes cleaned and reused. SEM stubs are cleaned. Often it's sufficient to manually scrape off the samples on the SEM stubs. For many of our specimens where you don't need to see the edges of the specimen, you are only looking at the surface view micromorphology, it is fine to even have a bit of debris from the previous sample on the stub. But if you need the stub to be perfectly clean, then acetone is great. If you soak the stub in that for a couple of hours to really clean the stub, and then use some scouring powder, normal dishwashing scouring powder to clean the stubs. And our technician in the microscopy lab is able to regenerate these stubs to look basically as good as new, as you can see on the right-hand side there in that central image. So everything that's possibly reused is regenerated. Chemical waste is, of course, always part of a lab. So both for limiting our environmental impact, but also for saving funding, when reagents are prepared, only the amount that is absolutely necessary for the experiment is prepared at any one time. All dangerous chemical waste is captured and labeled and stored safely in chemical storage cabinets. And when the storage containers are full, they are professionally removed, as is required by our university ethics. And then finally, go local. So we're down here in South Africa. Anything we use is imported mostly from Europe or from Asia, which has a big carbon footprint. It's transported over very long distances. So wherever possible, we make our own storage boxes, containers, even dissecting equipment. We use what is available locally or what is manufactured locally. For specialist equipment, we have a really great team at the instrumentation department, and they can basically build you anything. So we try to use locally made materials as far as possible in our lab, and of course to reuse that as often as possible as well. So as I said, there are some challenges. Everything is not always working as you intend, but every step you take towards a greener lab is a step in the right direction. Thank you.
Thank you so much, Lisa, for that enlightening talk. It really showed us, like, when presented with challenges, how we can make appropriate differences at an individual level as well as at a university level to overcome all those challenges and make our day-to-day research a little bit more sustainable. So now we have our last presenter for the day. So before that, I'll just quickly reintroduce myself. So I'm Aditi. I'm a postdoc at University of Pittsburgh. So here I am studying a lot on how receptor-like kinases, they are involved in plant immunity, and that involves a lot of like imaging and microscopy and generating tons and tons of data. So now that I have so much data, you always now in current climate, you have so much data and you're generating like terabytes of images and like during your entire project. How do you make sure that that is sustainable? Like make sure like you use the most of the data you have generated. So that's what our next speaker is going to talk about. Next slide, please. So yeah, Dr. Malia, she is a principal investigator at Donald Danforth Plant Science Center, and her research group actually focuses on how the mechanisms of crop resilience are like the entire mechanisms under these temperature stress. And to study that, she has developed an open source platform called Plantae. So I'm sure like most of you guys must have known and like used it in your research work already. And so she generates this entire like open source platform, and now she'll be talking about how do you make use of that data you have generated to more like you get more sustainable research out of all the data that you have generated. So over to you, Malia.
Great. Thank you, Aditi, for that introduction, and to Plantae for organizing this seminar. I've learned a lot. Okay, let me get those slides shared. So as Aditi mentioned, I'm going to focus my talk mainly on software data and a little bit on lab structure, and really with a focus on what my lab is focused on. So I'm at the Donald Danforth Plant Science Center. It is a nonprofit research institute whose mission is to improve the human condition through plant science. And we have two values. We have 5 values, but 2 values that are particular to this particular seminar, which is environmental sustainability, as well as stewardship of resources. And so when I say sustainability, for me, my definition of that is sustaining resources, sustaining efficiency, sustaining productivity, sustaining collaborations, but also sustaining work-life balance as well. So I want to take a brief aside on how I got here, because it is relevant to my focus on software and high throughput phenotyping. So I started working on high temperature and thermotolerance in guard cell protoplasts at a PUI. I then started looking at cold signaling and the relationship to the circadian clock as a graduate student at Michigan State. Temperature stress was really interesting, so I continued that work at the Donald Danforth Plant Science Center. And here is where I started a project called Plantae with a collaborator named Noah Falgren, who I continued to collaborate with and sustain that collaboration since I was a postdoc. And in my own group, we continue to use those resources. And I continue now to have to balance my interests in science along with my interests in maintaining that work-life balance with my family. So the theme of my science is really an interest in abiotic and environmental stresses. And one challenge when we study environmental stress over time is that much of the measurements are destructive. So every time you want to ask a new question, you have to do a new set of experiments, because we know that time and stress have a close relationship. So if you give a plant a stress treatment in the morning, you can get a different phenotypic response than if you gave the same stress treatment in the evening. If you gave a plant a stress treatment when it's a juvenile or a young plant in its vegetative stages, you could get a different result than if you were applying that stress when it's in its reproductive stages. The duration of a stress matters. So a long progressive stress is different than a short acute stress. And then the time in which you phenotype a plant matters. So if you give a plant a stress treatment and look hours or days later, you might see no phenotype. But then if you looked weeks or months later, you might see a phenotype. And so this means that we come against the problem of having to balance doing lots and lots of experiments and destructive measurements that are time expensive. And so all of us, when we have to collect data, one major bottleneck is measuring this variation, collecting this variation, and doing it efficiently. And so I'm lucky that at the center has invested resources into high throughput phenotyping technologies. So these are not I would not call these energy efficient technologies, but what I will say is that they allow us to collect more data per single experiment, and so that we can likely do fewer experiments in the long run because we're collecting that data in a single sort of set of experiments. We try to democratize the expense of these type of experiments with technologies like Raspberry Pi computers and cameras, so we can collect these types of data at lower cost. We use different imaging modalities, and we do this in different situations, like in the lab and in the field. So we're collecting lots and lots of data, and we need to analyze that data. And so the way that we do that is through the image data that we are collecting. And when you're doing this at high throughput, you can either do that perfectly, you can do that fast, or you can do that cheap, but you can't do all three. You usually pick two. And usually the traits that you're interested in are dependent on the question you're asking, as well as the species you're analyzing. And what we saw was that there are a lot of tools out in the community that were maybe very specific to a question or very specific to a species, but they were not reusable tools from project to project. They were maybe very specific. And so this is when we started to think about building a sustainable tool that would be modular and reusable, that could be combined so that you can ask different questions with different plants. And important to us is that the tools will be usable by both data scientists and biologists. We saw over and over again that interesting tools were being made, but that they weren't being maintained. And so we've sustained PlantCV for over a decade now, and part of that is because of the modularity of the package. And so again, PlantCV is a library of modular functions that you build into pipelines. You then parallelize that tool over a set of images, and then you get all of the data that you're interested in out once you've extracted it. We don't build PlantCV in a vacuum. We're not trying to reinvent the wheel. So we're interoperable with other open source packages. So that you can use those tools in combination with the tools that we build. So we spend a lot of time on sustainably developing PlantCV. We could have just sort of thrown software out on the internet and then hoped that was useful for other people. But we spend a lot of time on developing documentation so that it's usable by others. And then we spend a lot of time on the infrastructure of PlantCV so that other people can contribute to the code base. It is not sustainable to have a code base that can only be maintained by a single person. And so we have processes in place so that if I contribute something to PlantCV that breaks something that already exists, I can't combine those changes until those conflicts are resolved. And so with that infrastructure, we can accept community contributions and have the software be more sustainable. So PlantCV sustainability takes effort, and we are always trying to encourage people to contribute to not just our software, but any other open source software that has these sorts of frameworks in place. And that's important because if you are contributing to larger open source projects, you can have a larger community impact. So I'm going to use PlantCV as an example here. we, over our decade, have had about 38,000 downloads. We started with 3 contributors. We've now had over 61. There's been over 8,000 commits to that software base. And that's important because that means it's actively being developed, so that if you're a graduate student and you start using this tool, you want to be sure that if you learn the tool, you take the energy to learn that tool, it'll still be there when you're trying to graduate. And so for an important metric for us is that we have had 8 papers on the development of PlantCV, 119 user papers. So these are people that are using PlantCV for their materials and methods, and 35 thesis projects. As a relatively young lab, this is, of course, a much greater impact than that we could have publishing papers. These are vast majority of these are not our papers. These are the papers of others. And so this, again, because it's an open source, open development tool, is a tool that's used internationally, which is very impactful for us, which we love. And it means that people aren't having to maybe redevelop the wheel every time they need a tool. It's used in non-plant science research, which is really exciting for us. So these are not roots, these are cracks and sidewalks. So people in material science are using it to measure these cracks. People in animal science are using PlantCV to measure eye vessels. People were using PlantCV to measure the length of frog vocal cords. And maybe our weirdest example was there was a thesis project who was using PlantCV to measure the angle of flickering candlelight when they modeled historic sites. So this is a rendering of a historic site. So when we develop PlantCV, again, we're trying to do that not in a vacuum. So I'm going to give an example of two examples of how we use PlantCV and try to develop these tools in a sustainable way. And so we had a collaborator come to us with a set of data. This data was on wheat rust, and they were spending time having experts quantify this particular trait, giving it a score between 1 through 5. They had thousands of these images. At the same time, we were developing a naive Bayes classifier module in PlantCV, simply for segmentation. It was calculating the statistical probability that a pixel belongs into the plant class or the not plant class. So when they came to us with their wheat rust problem, we thought, oh, we can just modify this tool so that instead of 2 classes, we can now do multiple. So this same tool then we had used to categorize rust, chlorosis, and healthy tissue. And then we could give the researchers the % coverage of each of those classes. And so not only could they get a better quantification of that trait, but a faster quantification of traits. So we could analyze their full 1,000 or 2,000 data set in about an hour. So this is great. And again, this is a tool that we try to make sure that has multiple uses. So we develop these tools so that they're generalizable. So this is a different biological question here. We're now, instead of a disease question, we're using this same tool to categorize if the plants are stressed or not stressed. So this is an abiotic stress in this case. My second example is one that some of you might be familiar with. You might have used ImageJ to measure stomata openings from microscopy data. We developed a tool to make this more fast in a semi-automated way to count and measure the stomata apertures. We then used the same tool in a different way to measure pollen germination. And then the same tool is used in a different application. If I can change the slide, maybe, to measure the quantity and size of salt bladders. So same tool, multiple use cases. And there people are always showing us use cases that we don't expect. So you might not do high throughput phenotyping, but everybody, I'm sure, on this call probably generates data. And so again, I want to remind you that, especially if you're funded off of grant funding, all of the data that we generate, we should aspire to make that data fair, findable, accessible, interoperable, and reusable. And this can be challenging depending on the scale of data that you are doing. And so I just want to remind people that you can make data fair at different levels, and that you can make it fair to you, fair to your lab, and then fair to your community. And we can spend a lot of time on best practices on how to make data fair. But just some simple tips that should be hopefully obvious to many of you. You should give these files informative names. You should have a file structure that's logical. And you should make sure that, and this sounds like a joke maybe, but you shouldn't keep all your files in a single folder, but we've run into this case. You should not keep your folder, your files in the trash, which was another case that we ran into. And you should make sure that you can find the files that you need. So this isn't a snapshot of my electronic lab notebook. I like to have a list of tasks that I do per week. And then I, in this notes column, put where the files are that I've been working on. And since this is a Google Doc, I can search that. So just as a reminder, so if you won the lottery today, would your lab know where all the data you have is with experimental data? Can you find, can your lab then find where your data is, even if they know that you have it? Is your data accessible to your lab? Is it in the cloud, or is it on a laptop without a backup, which is not the ideal use case? Is your published data accessible to the community? Can your figures and analysis be easily reproduced? And do you include enough metadata for your community to use your data? Are you using all the tools at your disposal to make this efficient? And just a reminder, this does not have to be perfect. You can improve it as you go along, and it is definitely not easier to do it tomorrow. And so I'm going to just spend 1 minute on some sustainable lab structures, because I mentioned that I started this PlantCV collaboration with NOAA back when I was a postdoc. And we were both, it was a sort of an unusual case that we were both able to stay at the Donald Danforth Plant Science Center. And so we are at the institutions that we did our postdoctoral research at. And so we've sustained this collaboration, which has been very influential on my own career. And so my lab is a lab of really 8 awesome, really awesome and collaborative people. But we do this in a framework that's much larger. So NOAA and I have shared researchers between our groups, and then Katie Murphy, who runs our phenotyping core, we also have shared people between our groups. Why this is, to me, a sustainable lab practice is because that means that in terms of mentorship, a lot of our folks in our lab have dual mentors, and even the people who are not mentored by my lab know that they can go to NOAA for mentorship, maybe when I'm not available. And so with that, I'll just reiterate these take-home messages. So green lab practices can take time and effort up front, but hopefully will save you time and effort in the long run. Use existing tools wherever possible. Use resources like GitHub and public repositories. Document, and use version control if possible. Make data fair, and it's important for you as well as your community. Use community standards. Electronics die, so don't store things on local computers. Form a circle of trust and build a village to help you make your lab practices more sustainable. And done is better than perfect. And so with that, I will hand it back to our awesome Plantae Fellows for the question and answer session.
Thank you very much, Dr. Malia, for that amazing talk. It was so interesting to see the emphasis that you put on community and how important it is to become more efficient with the data that you're handling. I thought that was a really interesting perspective to have. And thank you to all the speakers for your amazing talks. It was great to hear from all across the world and practical things that we can put in place in order to become more sustainable researchers. So we have one question, and I'll direct this to Caitlin, our first question. And this is, how can researchers combat struggles with the time and resources for managing things like the freezer maintenance that you were discussing? And whether you have any recommendations for this?
Yeah, it's a hard one. Everyone's very busy. There's a lot more things we all have to kind of do. We don't necessarily have as much lab support to kind of take care of things that might have previously been done by technical staff. I guess nothing's going to make the day longer than it already is. So if we can kind of integrate it with practices we're already taking part in, that's going to be the best way to do it. So for example, I don't know, yeah, scraping ice off a ULT freezer. If you just have an ice scraper next to the freezer or in the room, you've opened the door, you just do a quick scrape. It's not necessarily something you need to do every day or every time you go in, but at least if people have started doing it, it's going to make a difference. Things like, you know, 3 monthly filter cleans, it takes about 5 or 10 minutes. If you have a rota and you really split up that responsibility, it also means your freezer is going to last longer. You're not going to have to buy a new one. So it's kind of finding the balance between those extra tasks that are more sustainable, but also, like Malia said, it's going to save you time and pain in the future if you kind of put a little bit more into your daily activity now. Some of the large things like washing up and reusing glassware and plastics instead of buying single-use plastics, over time it will save money. So you could invest that money into having a person to help you. But in the first instance, it might be that someone who's really keen and really passionate and maybe has half an hour can just kind of trial things and see how it goes. And then you could write a business case and kind of put together the financial benefit, because it's all the goodwill in the world saying we should be doing this for the environment. But again, you know, researchers, that's not always the priority. Finance and time are two big barriers. So if we can show it's going to save money, then we might be able to get a resource to help us save the time.
Great. If anyone else has anything they wanted to add to that, then please feel free to jump in. If not, then I'll move on to our next question, which is directed at Dr. Malia. This is referring to whether there is any imaging software that you can recommend which relates to stress-related hormone identification. That's in the chat.
Sure. So usually what we're doing is looking for, in terms of images, if you're looking at the whole plant level, we're looking at proxy traits related to maybe stress hormones. So, you know, if you have ROS production, are you impacting the color of those plants? And so often you can find the downstream effects of those stress hormones with other traits when we're looking at image data. If you're actually specifically tagging some of those stress compounds or metabolites, you know, using microscopy, then, you know, we can help with the, or other software packages can help with the analysis of those data to make those things more efficient.
Great, thank you so much. I think as we're running a little bit over time, if everyone's happy, I might round the session up. But I'd just like to thank all of the speakers and thank all of the audience members as well for your participation. It's been a really fantastic webinar and I've really enjoyed moderating it. And I'll pass it over to Gabriella for the final points.
Sorry. Hi. Thank you to all the speakers for bringing us your insights from all over the world. So different scenarios that we as plant scientists can face, and to learn that we are concerned and we are making decisions based on our role on Earth and on sustainability. I'm pretty sure we could have learned more about, for example, how we make decisions on our field work, on our travel. When we go to a conference, it's always a struggle, like thinking about how much carbon emissions we're going to have by flying around, or just like maybe like Lisa, like going around South Africa, investigating plants, stuff like that. So any other questions the audience might have, feel free to email Jason or to email us. I want to thank my co-fellows, Kes and Aditi, for working together in this, to our great speakers, to ASPB and Plantae for giving us the opportunity to organize this webinar. And I'll give the last word to Jason to close the webinar.
Great, yes. So I was just going to echo what Gabriela said. A big shout out to all our speakers today. I've learned myself so much with everything that you've shared. And thank you for your energy and all the great work that you're doing in your spaces to help make labs green, and also sharing those knowledge to the rest of the plant science community around the globe. And thank you, and a big shout out, of course, to our Plantae fellows who spent so much hours getting this together and organizing this webinar. And to all our audiences, thank you for tuning in, and we hope to see you in the next one. Have a great weekend, everyone, and hope to catch you again on our next webinar next month. Have a great weekend, everyone. Bye.